Hello, everyone. Welcome to today's APS respiration section webinar, the title of which is Heterogeneity and Acute Respiratory Distress Syndrome. I'll be serving as the moderator. As many of you know, I'm Chris Waters. I'm a professor of physiology at Augusta University, just joined in the last month and a half. I'm also the chair of the APS respiration section, which has the section that organized this webinar. In today's session, Dr. Michael Matthay and Dr. Narges Alipanah-Lechner are going to discuss clinical and biological features of the acute respiratory distress syndrome. And they're going to be emphasizing the importance of metabolic signatures and the relationship to hyper and hypoinflammatory phenotypes. I want to point out a few housekeeping notes. Out of respect for the speakers, we ask that you refrain from photo or video captures of the presentation in whole or in part. The session will be recorded and available for future viewing through a link on the registration website. Taking photographs of another person's presentation without their explicit permission is prohibited. At this time, all attendees except for the speakers are in listen-only mode with the video off. As the moderator, I'll be happy to raise questions that are entered in the Q&A function on the bottom of your screen. So if you have a question during the talk, and we're going to have a question and answer session at the end, just put it in the Q&A thing at the bottom. Before we begin, I'd also like to thank today's sponsor, LSF Medical Solutions, for their support of our section's awards and social activities. In addition, I really want to make a special request to all of you out there to provide support for the APS Charlie Bates Award in recognitions of scientists of the LGBTQIA+ community. There's a URL listed on the side, and there will be one in the chat if you want to make donations for that award. The session will end at approximately noon Eastern Time. And now I'm going to turn it over to Dr. Usha Raj to give the introductions. Dr. Raj is from the University of Illinois, Chicago. Usha, you're up.
Okay, well, thank you. Thank you, Chris. And I want to also thank Melissa Bates, the owner of the company that's sponsoring this webinar, and who also very kindly asked me to introduce Michael and Narges. So I will just say this is absolutely such an honor to introduce my very dear friend, colleague, mentor of many years. And I think a lot of you know him and know of him. And you're going to meet Narges today. So next slide, please. So I want you to look carefully at this picture. You see this young man. Notice his gaze. He's so confident. He's looking straight at you. And also, don't forget to notice how well-dressed he is and how he attracts people all around him. And that's his personality and has remained that way to this day. So next slide, please. And this young man is brave, fearless. He's riding the waves, and he has shown that same quality when he goes into the lab trying to uncover mysteries about ARDS and other diseases. Next slide, please. Now, this young man did get a very good education, went to Harvard, UPenn, Colorado, but his research training, which I think he would agree has created this person who can do such wonderful research that happened at the Cardiovascular Research Institute. And I have to mention his two mentors, Norman Staub and John Clemens, among many. But I have to say this. His years in Boston at Harvard University completely spoiled him. He is just such a diehard Red Sox fan. Oh my gosh, can I have the next slide, please? And every year he goes, and now he goes with his two grown sons to fantasy camp to play baseball with the Red Sox. So, I mean, kudos to you, Michael, and I know you won one of these matches one of these years. Okay, next slide, please. Now, why am I so proud and so privileged to introduce Michael? Well, he's just outstanding in all the areas that I've listed here: bench researcher, clinical investigator, clinician, educator, and most important, I think, as a mentor and sponsor. And a word about that later. Next slide, please. He has won numerous awards. I mean, just numerous. I just highlighted some of them. The very first one, ATS Clinical Care Assembly, that just shows just how much committed he is to clinical medicine. The mentoring awards that are just amazing. He's got one in 2013 and one in 2021. Then the Livingston Trudeau Medal, which is literally the highest honor that ATS gives. He got the Julius Kammerer Award, which is the highest honor that the respiration section at APS gives. Next slide, please. And has won many other awards too. I just want to say that his work, and this is remarkable, has been funded continuously for over 45 years with funding from NIH, with R1 grants, PPGs, multi-PI, huge clinical studies, and he goes nonstop. Whenever I try to reach him, he's busy writing a grant or editing a paper. Next slide, please. So all those years of research have generated important information that have helped us understand these 5 areas. This is where his research has been focused. His over 1,000 original papers, chapters, editorials, commentaries. But the biggest, I would say, I'm only put ARDS on the top because he's going to tell us about that. But he clearly, both in the lab and in the clinical setting, has done innovative work. And I think he was one of the first people who started to use human lungs to do research in the lab, and it's given us information that is so clinically relevant. And that, of course, as I said, fundamental studies in ARDS. His very original work was on figuring out how fluid and protein is cleared from the alveolus. And that, of course, is very relevant when we try to understand how patients with lung disease can recover. And he has been the promoter of large clinical trials, and one of the very original studies which showed that increasing PEEP improved survival, which was a big breakthrough because for years we couldn't find anything that improved survival in ARDS. And that original clinical trial was something that Michael led. And he's going on to do more innovative stuff with mesenchymal stem cells and experimental and clinical RDS. Okay, next slide, please. Now, I do want to obviously introduce your second speaker, Narges Alipanah-Lechner. And why is she important? Because she also represents what Michael does. Michael mentors and he sponsors. His has mentored so many people. He has particularly, I would say, helped bring up young women who oftentimes are left behind. And starting from Janine Cronish and Lorraine Baer and Carolyn Calfee, and now Narges. Narges is almost like the second generation because Michael trained Carolyn Calfee, and now she's being trained by both Carolyn and Michael. And she clearly is a star, you know. And why do I say that? Because Michael thought about including her in this presentation, but I think this also highlights his quality, which is he shares, he mentors, and then he sponsors. He will invite people to events like this so then they can jump off and continue their career. And I have no doubt that Narges will continue along the same track as his previous mentees. So at this point, I want to turn over the stage to Michael. Michael, we're all waiting to hear what you have to say.
Well, first of all, that was an incredibly overly generous introduction, Usha. Thank you very much. You know, I think we all know it's working with each other and locally team science and others. So these are all shared accomplishments. You're very generous, but thank you. But I just appreciate it. But it's a group effort. Thanks very much. And so let's see, I'll try to start my slides here. As already mentioned by Chris and Usha, this will be a two-part presentation. I'll try to focus in on some key issues in ARDS, but then leave the last half for Narges to provide some very interesting new in-depth studies of mechanisms that I think are very important for pathogenesis of ARDS, and there might be a time in the future when would translate to therapies. So ARDS, clinical and biologic heterogeneity, disclosures, no conflicts for this presentation. So ARDS, everyone I think probably knows the original report was by Ashbal, Petty, and others in the Lancet in 1967 of a syndrome of acute respiratory failure with bilateral pulmonary opacities, pulmonary edema, cyanosis, and associated with trauma or viral infection or bacterial pneumonia. And so just fast forwarding to an overview now, this slide is intended to show a typical chest radiograph on the left, and then on the right, you can see the protein-rich pulmonary edema fluid in the alveoli. We think that ARDS occurs in about 200,000 patients annually in the US. It's defined by a PaO2, FiO2 less than 300 millimeters of mercury, mortality depending on initial severity, 25 to 45%. Interestingly, the incidence went up tenfold during the COVID pandemic. Main etiologies are bacterial and viral pneumonia, sepsis, aspiration, and trauma. The latter less than it used to be because of a better resuscitation strategy. And the treatment with lung protective ventilation, prone position, and fluid conservation strategy have really improved clinical outcomes from classical ARDS. It's just very fortunate that we have had those therapies during COVID, or the mortality from COVID would have been much worse. And I'll talk just very briefly about pharmacologic approaches. We don't have much. That's a big part of the theme of this talk and why we need more basic research in terms of pharmacologic therapies for ARDS. Now, I want to mention some of the pioneers that really helped our field. And Norman Staub, whom Usha has already mentioned, was my primary research mentor. He's really the father of pulmonary edema. And it was Norman who took Starling's 1896 equation that quantifies fluid movement across the semi-permeable membrane to define the mechanisms explaining the pathogenesis of pulmonary edema. There's quite a bit of confusion until Norman did this work. And he made it clear that there's a hydrostatic edema from an increase in pulmonary vascular pressure, and increased permeability edema from an increase in lung endothelial permeability. Norman devoted much of his life to APS. He was chair of the respiration section, and he was the reason I got involved in APS originally. And just to have a quick clinical translation of what Norman was writing about and studying experimentally, and that is that in patients who are intubated, there are two films here, the one that you can see on the left and the one on the right. You can't really tell by looking at the film whether this is pulmonary edema from high pressure or increased permeability, but the pulmonary edema fluid on the left had a protein concentration of 3 grams, while the plasma was 6.1, so that's a ratio of 0.48. That's hydrostatic edema. And on the right, it's a high protein, 4.9, with a plasma of 5.5, a ratio of 0.89. So this was really important for the field, and follow-up studies that Lorraine and I did explored this more, but this was really an initial study in which Norman was a senior author. And then just a quick word, this incredibly important study, 1977 by Marianne Bachhoven and Ewell Weibel, established histologically at the light level and at the ultrastructural level severe ARDS in the lung and what it looked like. So it was a lot of edema fluid, inflammatory cells, those hyaline membranes in here, although they're not necessary to make the diagnosis, and the ultrastructural showing injury to the endothelium and the epithelium, which is denuded, and the attempt of the type 2 cells to recover the injured barrier. Now, just to sort of put this in context in sort of a visual way, this slide is intended to show a normal alveolus on the left and then the injured one on the right. And the main point I think to remember in ARDS is no matter whether the lesion is pneumonia, aspiration, or non-pulmonary sepsis, the primary problem is increased vascular permeability, to some extent almost always mediated by neutrophils and platelets, and then injury to the epithelial barrier with increased permeability. And then, as I'll mention, as Usha had talked about, impaired capacity of the lung, the alveolar epithelium, to remove edema fluid. And that's the project that I'll just share with you briefly, what she alluded to. It was Norman who said to me, you know, Michael, we don't really understand this. How is edema fluid actually cleared from the lung? So to explain this, we did a series of studies in sheep, and we found, somewhat serendipitously, that alveolar fluid clearance was driven by vectorial sodium transport. And we had instilled into the right lung 100 mLs of autologous serum. And we found by gravimetrics that 25% was removed in 4 hours using the contralateral lung as a control. But the key finding was the alveolar protein increased from 6.5 to 8.6 grams, much higher than plasma. And a series of studies, including apical sodium channel inhibitor with amiloride, wabane, and other studies showed it was sodium transport that was driving this process, and cyclic AMP agonists could upregulate fluid clearance, including in the ex vivo human lung. And then working with Lorraine Ware, who was a fellow with me in the late 90s, early 2000, we found in patients, using that same principle of sampling edema fluid right after the patient was intubated, and then 4 hours later, that the patients who had minimal clearance of alveolar edema fluid in green versus those with maximal clearance had a big difference in their outcomes. So it kind of provided a functional physiologic evidence to go with that study by Miriam Bachhoven and Weibel, that the alveolar epithelium injury, to the extent it occurred, correlated quite well with how bad the patients would do. And in several subsequent studies, we found that the mechanisms to explain this impaired fluid clearance included hypoxia. Just regular moderate hypoxia stops sodium uptake by the ENaC channels. High tidal volume injures the epithelium, pro-inflammatory cytokines, and also just epithelial cell death. So then I want to come back to Norman Staub again, because Norman had done these studies actually before I joined the lab, but some additional, in which in the sheep model, he showed that lung edema formation on the y-axis, as reflected by lung lymph flow, would increase as left atrial pressure was raised. And you can see this line here, sort of a linear increase as pulmonary hydrostatic pressure in the lung was increased. But if the sheep had endotoxin or bacterial injury, you can see, of course, that the lymph flow, edema formation, was already increased, but then just a small increase in vascular pressure, 2, 3, 4 millimeters of mercury, you can see the, a big increase in edema formation. And so 30 years later, this concept was translated to ARDS with the full fluid and catheter treatment trial that showed that patients who were given a fluid conservative strategy with ARDS had more ventilator-free days. And this is the results of that trial. It's one of the most important ones in our field, and it really goes back to the basic physiology, careful studies that Norman Staub did. And this study, as I said, showed the reduced duration of ventilator support with fluid conservative strategy. Also showed that PA catheter was not necessary in these patients usually. Then the other basic physiologic study that was done also here at UCSF in the CVRI, a very famous study by Webb and Tierney in 1974, which was overlooked for several years. But in rats, they basically showed, as illustrated in this image, that here's the normal lung in the rat, 14 centimeters water of inflation pressure, airway pressure. Then when they increased it to 45, you can see how the lung becomes hemorrhagic and edematous. But if they added tinapeep, thus reducing the inflation pressure, there was much less. So this is really the original trial that suggested high tidal volume, high airway pressure would cause lung injury. A lot of studies by Parker from APS in Alabama followed up with this, and Dreyfus in France. And then here's the study I think that Usha was referring to. This was the ARDS Network's first study, and it showed that mortality was reduced from 40 to 31% in patients with ARDS who received a lower tidal volume and a plateau pressure limit. And then I want to transition here to the theme here we're trying to emphasize, which is biology as well as physiology. So it turned out, fortunately, that we collected plasma samples in this ARDS trial, the ARMA trial, the low tidal volume trial, at the baseline and at day 3. So when we measured the levels of IL-6, IL-8, TNFR1, SPD, and RAGE being a marker of alveolar epithelial type 1 cell injury, in all these cases, they were markedly reduced in the patients who received the low tidal volume. So we then had a mechanistic understanding of why low tidal volume was working. That's in the plasma. And in fact, this is just an image, a figure to try and illustrate this. I did this study with Lorraine Ware and the late Guy Zimmerman, and you can see the injured alveolus here in panel A with the edema fluid. See the meniscus here, the neutrophils, the macrophages, injured endothelium and epithelium. High stretch, like we ventilated patients for 30 years or more, just exacerbated the injury, made more injury to the endothelium, epithelium, activated macrophages more, much more inflammation, and impaired alveolar fluid clearance. And then with low stretch, however, all these mechanisms were reversed. But this shows the entree, I think, of biology into the physiology in ARDS. And then one more study I just want to mention briefly is the study that was stimulated by Gattinoni's work, actually not JAMA 93, it should be Anesthesiology 91, but the bottom line, it was Gattinoni who published this paper that showed in 10 CT scans the patients with ARDS that their pulmonary opacities were movable. So when they turned prone, you can see how the consolidations went in the opposite direction. So it turns out a dorsal inflation of the lung, which is a greater mass, recruits more air spaces. There's a whole story here, but the bottom line is it was translated to a clinical trial by the French showing that prone markedly improves survival. Okay, so in 2019, just before the pandemic, several of us, including Carolyn, published this review trying to summarize a lot of the mechanisms we understood about injury, which are, significant: impaired fluid clearance, disrupted endothelium, epithelial cell death, and a whole variety of pro-inflammatory factors, and a moderate amount that we understood about repair. Of course, that was 5 years ago. There's more that's been learned since then. But the big problem is not much has been achieved to translate these mechanisms to new therapies. And so this is roughly where we have been for the last few years. ARDS is heterogeneous in so many respects. And so we've been working hard on this idea, as have others, on how to focus treatment strategies that focus on the heterogeneity. Just to give you an example, here are 2 chest x-rays of ARDS. They look pretty similar, right? Intubated, ventilated patients. The one on the left is vaping-associated ARDS, and the one on the right is COVID. And so we have this issue. In fact, it's the same issue John Murray and Tom Petty used to debate. Should we combine ARDS, lump it into one, or break it down? Well, for those ventilator trials, for the FACT trial, for the ARMA trial, and the PRONE trial, it was actually better to do a lot of lumping, although the PRONE trial used prognostic enrichment and focused on patients with more severe hypoxemia. So there's merits to both, but we think that the way forward for part of our work needs to be on breaking this down. And so there are so many different ways to split ARDS. Of course, there's COVID-19, more typical ARDS, direct versus indirect, patients who have persistent and rapid resolution. The radiographic pattern can be diffuse or focal. Physiology, high or low compliance. microbial cause, very different pathogens, virus, different bacteria. And so the issue is, how could we go forward? So it's now been 10 years since we published this paper. I think most of you probably know of this work. Carolyn has led this. The whole idea was suggested to Carolyn and me as we were crossing the street from the library when Kevin DeLucci, our statistician, said, "Michael and Carolyn, have you guys ever heard of latent class analysis?" We said, "What's that?" He said, "Oh, well, it's an unsupervised approach that can identify different classes that you can't see otherwise." So the long and short of it is, we gave him these clinical and biologic variables, and 2 weeks later he said, "Come see me. I think I have something interesting." And what turned out is that in the first study, the latent class analysis turned out to break down patients into a hyperinflammatory group, 1/3 of the patients, and the other hypo. Now, they're not really hypo, they're just less inflamed. And you can see on the left that what really put them into these categories in the case of hyper was plasma levels that were elevated for IL-6, IL-8, TNFR1, and PI1, or on the right, lower levels of bicarbonate, protein C, and systolic blood pressure. And many of the other factors that we used to classify patients were not important, the level of PEEP, the bilirubin, the age of the patient. And then it turned out briefly in subsequent studies over the past decade, this categorization has led to all kinds of secondary studies, including a pediatric group, the RESTORE study, that showed that the hyperinflammatory have about twice the mortality of the hypo. So it seems to identify a group that has a very different outcome, much worse. And then there's some reason to think that maybe this could be important treatment-wise, because in these secondary analyses, in one of the studies, there was a different response for high versus low PEEP, and the other one, the FACT trial, a different response for conservative versus liberal therapy. And then this one was simvastatin. Danny McCauley published a paper in the New England Journal of 540 patients treated with simvastatin, and there was no difference for placebo versus ARDS. But when we then subsequently broke these down into hyper and hypoinflammatory based on TNFR1 and IL-6, we found there was no difference in the hypo group, but the group that had the simvastatin in the hyper group appeared to benefit. So it suggested that maybe there'd be a treatment responsive subgroup. And then Pratik Sinha, who trained here at UCSF, now an independent investigator at WashU in St. Louis, he did a terrific job in breaking this down to a parsimonious set of variables. So we didn't have to use the latent class and 36 variables. And you can see with a 3 or 4 variable model, it turns out we have very good AUC for predicting hyper versus hypo classification, why does that matter? Because then we can translate that to the clinic, to the patients in the ICU. So right here in my lab, you can see we have this ELA made by Protein Simple from Biotechnique. And this will measure IL-6, TNFR1, IL-8, and Ang2 in 70 minutes, actually 20 to 30 microliters of sample. And we validated it in several studies, 2 of them listed here. So because of this ability, we just completed an observational study led by Clark Files at Wake Forest, in which we have shown in over 300 patients that we can classify patients with ARDS rapidly with this method. So therefore, Danny McCauley has received funding in the UK to carry out a phase 2 precision medicine adaptive platform trial in which we will use real-time measurements in the patients to classify them to hyper, hypo. It's very exciting. It's going to start next year. And currently the treatment arms will be simvastatin, baricitinib, because baricitinib, JAK2 inhibitor, worked modestly in the pandemic. And we've applied for Department of Defense funding to fund this trial also in the US. So we're very excited about this. It'll be phase 2. It has a Bayesian plan for the statistical analysis. And so this could translate to maybe finding a method to classify patients and find which ones would be treatment responsive. So in summary, before I hand this right over to Narges, ARDS is a major cause of mortality and longer-term morbidity. Effective therapies, as I briefly reviewed, are low tidal volume, fluid conservative therapy, and prone positioning. I think inspiring examples of the success of translational science. Efficacy of pharmacologic therapies is limited for ARDS except steroids for severe community-acquired pneumonia without shock, and some evidence, as most of you know, for COVID-19. But we're looking to have predictive enrichment with these parsimonious biological markers and clinical markers that may identify treatment-responsive patients. We need more basic insights here, however, into pathogenesis to generate new therapies. Hence, what Narges is going to present to you. And I just firmly believe that continued basic and clinical research is needed to fuel new therapies, and training the next generation of clinician investigators is the highest priority. Why don't I stop there so Narges can take over. Wait, I gotta hit stop share here. Okay.
All right, thank you so much, Michael. Let me just share my slides. Share. Are you able to see this okay?
Yes, looks good.
All right. Hi, everyone. I'm Narges Alipanah-Lechner. Thank you so much for the invitation to be here with you today. I'm excited to show you a little bit of what I've been working on with Dr. Calfee and Dr. Matthay, and get your thoughts on sort of where we should go with this data next. So I'll just briefly touch upon the inflammatory phenotypes that Dr. Matthay already discussed, since I'll be giving you a deeper dive into their differences in just a few minutes. So Dr. Calfee and her research team conducted a secondary analysis of now over 7,000 patients with ARDS from several randomized control trials and observational studies. And they specifically took clinical data at the time of study enrollment, together with a set of protein biomarkers that we think are associated with underlying pathways of injury in ARDS. And they combined all this data together, applied latent class analysis, which is a type of cluster analysis, to see if patients would separate themselves into unique clusters. And indeed, they did. So over and over again, in each study, patients seem to fall into these two buckets that they labeled hyperinflammatory and hypoinflammatory, with the hyperinflamed patients being about 1/3 of study cohorts typically, and having over twice the mortality rate, which is not a subtle difference clinically. And these are the trials and observational cohorts that Dr. Matthay already showed. And as you can see, the mortality rates can be as high as 60%, depending on the study. And in 3 of the studies, again, as Dr. Matthay mentioned, there was evidence for heterogeneity of treatment response based on the inflammatory phenotype that the patients were assigned to. So these phenotypes were independently confirmed by other investigators as well, from other institutions in the US and Europe, as well as 3 pediatric ARDS cohorts. And our research group has observed the same phenotypes in secondary analyses of sepsis trials as well, suggesting that these phenotypes may not be specific to ARDS, but actually traits of critical illness. And so what do we know about the biology of these phenotypes so far? This is the spectrum of genome to phenotype. Our group has studied biological differences between these phenotypes at several of these approaches. I mentioned that the phenotypes were originally derived using protein biomarker data, and Dr. Matthay already explained that that data demonstrated that there was increase in circulating biomarkers of inflammation, disordered coagulation, endothelial injury, and alveolar epithelial injury. Now we get to my area of focus, which is the metabolome. And I'm interested in the metabolome because on this spectrum, it's closest to phenotype. So presumably it's the most correlated with what we're seeing clinically. And tied into that concept is that metabolites in the cell can change quite rapidly to environmental stresses and cues. And so the metabolome sort of gives you a snapshot of gene function, enzyme activity, sort of what the patient is receiving all in one at one time point. And of course, the most widely used biomarker in critical illness is lactate. So there is hope that perhaps by delving deeper into the metabolome, we can better understand mediators and pathways involved to then hopefully intervene upon them. And my co-mentor, Dr. Angela Rogers from Stanford, together with our UCSF research group, published this interesting study back in 2021 to answer the question of whether metabolic profiling could identify patients with early sepsis. They used principal components analysis and PLSDA, hierarchical clustering, and they were able to identify 3 subgroups of patients, as you can see from the PLSDA plot and the heat map here, with very distinct metabolic profiles. And interestingly, patients in group 1, which were about just over 20% of the cohort, had a significantly higher 60-day mortality rate. They also had higher rates of vasopressor-dependent shock, higher rates of kidney injury, and conversely, group 3 seemed to have a more favorable phenotype, and group 2 was somewhere in the middle. In this study, they were not able to identify a particular metabolic signature that distinguished those with ARDS from those without, though there was a higher likelihood that patients with ARDS belonged to a group 1 phenotype. So based on that data and Dr. Calfee's work, I became curious to see whether the LCA phenotypes of ARDS exhibited different metabolic profiles. So in my first analysis, I used a subset of patients from that paper that I just showed you by Dr. Rogers from the early observational cohort. This is a cohort of critically ill adults admitted from the emergency room to the ICUs at 2 San Francisco hospitals. And physicians later adjudicate the presence of sepsis and ARDS by reviewing the patient's charts extensively and the entire hospitalization to determine whether sepsis or ARDS was actually present or not. So a very well phenotyped cohort. Within that cohort, I selected those that were deemed to have sepsis, and then out of those patients specifically, I restricted my analysis to those who had ARDS. And then we phenotyped them using latent class analysis into hypo and hyperinflammatory phenotypes. And then we used their plasma samples at the time of study enrollment to perform untargeted metabolic profiling. This was done by Metabolon, and see if we find any metabolic differences between these two. And as expected, this was a small study, as you can see by the numbers, but the characteristics of the patients differed by inflammatory phenotype. So hyperinflamed patients were more likely to have shock and higher sort of scores representing disease severity, higher rates of acute kidney injury, and significantly higher mortality. And then I used linear models for microarray data, or LIMA, which we commonly use in transcriptomic analyses, and applied it to the metabolomic data set and adjusted my analyses for sort of confounders that we typically think of as impacting circulating metabolites. So age, sex, use of some ICU medications like propofol and dexmedetomidine, as well as liver and kidney disease. And after that, we found that 82 metabolites were significantly different between the phenotypes. So here, this heat map is clustered by phenotype. And as you can see, there are a lot of metabolites here that are lower in the hyperinflamed phenotype. And I would say this is basically over 90% of the lipids that were significantly different between the phenotypes were lower in the hyperinflamed patients. So this is the volcano plot showing you the log 2-fold change in metabolites. So metabolites in red are higher in the hyperinflammatory phenotype, and the ones in teal are lower. So lots of lipids are lower in the hyperinflamed patients. Instead, the hyperinflammatory patients seem to have higher levels of metabolites like lactate and pyruvate, which we sort of interpreted this as suggesting a glycolytic shift towards lactate production. So putting all of that together, we came up with this potential mechanism for the signals that we were seeing, that perhaps dysregulation at the level of the mitochondria might be leading to this sort of shift towards lactate production and a drop in lipid synthesis. We then set out to determine whether this metabolic pattern was just sort of a spurious finding from this observational cohort or more a consistent feature of the hyperinflammatory trait. So for the second study, this is unpublished data that I will be showing you that we're still working on completing. But we used data from the ROSE trial of neuromuscular blockade for the treatment of moderate to severe ARDS. And this was another large, well-designed, but negative trial that enrolled patients who had severe ARDS as defined by P to F ratio of less than 150. Patients were randomized to either receive neuromuscular blockade, and this was cisatracurium with deep sedation or light sedation in the control arm. And the primary outcome, which was survival to hospital discharge, was not different between the two groups. Dr. Sinha and Dr. Kalfi had previously completed latent class analysis phenotyping of this cohort, so we had data available on who is hyper and hypoinflammatory in this data set. Out of the entire data set, we randomly selected 160 patients with the specification that they must have had a high probability of belonging to either the hyper or the hypoinflammatory phenotype. So a probability of greater than 90% that they would be one or the other. And we selected 80 patients from each phenotype to have adequate power to really maximize our ability to detect differences between these two phenotypes. We had day 0 and day 2 specimens on these patients. Whole blood transcriptomics was done, and then we added untargeted metabolic profiling as well, using these 3 different platforms that, again, was performed by Metabolon. Lipids were the predominant class of metabolites that were identified, as you can see, followed by a lot of metabolites that are unknown. So I excluded the unknown metabolites from my analyses. And here we actually observed a much stronger signal of metabolic differences with over 500 metabolites that were distinctly different between these two phenotypes. Here's the volcano plot. Lots of metabolites are higher in the hyperinflamed phenotype. And again, many lipids seem to be lower. I then performed differential abundance analysis, again using lemma, adjusted for the same confounders that I had mentioned previously, and this time I actually included glomerular filtration rate. And again, you see that across almost every metabolic class, the metabolites are going in opposing directions by phenotype. I then performed enrichment analysis to see what pathways come up significant. And not surprisingly, based on that heat map that I showed you and the number of metabolites that were significant, I got over 40 pathways that were different. But for the purposes of this talk, I will focus on a representative few. So at the top, we see that there's decrease in long-chain acyl carnitines, both saturated and polyunsaturated, and indicating potentially impaired fatty acid transport into mitochondria. And it seems that cells are then sort of trying to use perhaps alternative pathways for energy production. And we can see there's increase in carboxylic fatty acids in particular. These fatty acids are generated through a process called omega oxidation through the endoplasmic reticulum. And this process sort of takes over when the usual beta oxidation through like the normal mitochondrial fatty acid oxidation pathway is either impaired or overwhelmed. There's an increase in lactoyl amino acids, and that suggests that pyruvate is perhaps being diverted from normal energy metabolism to such high levels of lactate that lactate is just binding amino acids as sort of potentially a buffering mechanism. There is a increase in medium chain fatty acids, elevation in TCA cycle intermediates, again suggesting that perhaps TCA cycle isn't being completed and generating sort of these partial metabolites. There's increase in glycolysis, gluconeogenesis, suggesting that the cells are really trying to maintain energy production. Elevations in leucine, isoleucine, and valine metabolism sort of suggests there's increased protein breakdown for perhaps the body's trying to mobilize alternative energy sources in addition to lipids. And then changes in nicotinamide metabolism as well suggest that perhaps there's an altered redox balance. So every pathway seems to kind of tell part of the same story, that maybe the mitochondria can't properly process fatty acids. They're trying to perhaps rewire their metabolic network to maintain energy production in a setting of really high energy demand. Then I set out to determine if sort of adding whole blood bulk transcriptomic data, which I mentioned we had available in this cohort, to metabolomic data would add any new insights. And to do this, I used this bioinformatics tool called multi-omics analysis, or MOFA. This is sort of an unsupervised factor analysis that takes concepts from principal components analysis. It basically, if you have multiple data modalities, so in my case, transcriptomics and metabolomics on the same patients, MOFA tries to find coordinated axes of variation that explain the heterogeneity or variance in a data set. So to do my MOFA analysis, I restricted the data to the top 500 metabolites that had the most variance in the data set, and the top 2,500 gene transcripts that also had the highest variance. And again, this is an unsupervised approach, so there's no clinical data that's included. I did not include protein biomarker data or LCA phenotype designation. So this is just clustering metabolites and genes and asking this model to generate or decipher sources of heterogeneity. And it turns out both metabolites and genes explained a decent amount of variance in the data, so with genes having a slightly higher contribution. And then these are the latent factors that MOFA picked up. Each factor, again, is a combination of genes, gene transcripts, and metabolites. And as you can see, factor 1, it seems to be mostly explained by genes. Factor 2 is mostly explained by metabolites. Factors 3 is a combination of both. And then after that, there seems to be little contribution or very little of the variance in the data set that's explained by these data types. This plot is showing you the association of factors with clinical variables. So after running MOFA, I added clinical variables to see if the factors were clinically meaningful in any way. And so the x-axis has the variables that I tested, and the y-axis is all the factors that MOFA had identified. And the colors correspond with the direction of association, and the size of the dots corresponds with the significance or the strength of the association. So as you can see, the first 3 factors, which I mentioned were the ones that explain most of the variance in the data set, are all 3 associated with LCA phenotypes. And interestingly, all 3 are also associated with disease severity, and this is Apache 3 scores. But only factors 2 and 3 are correlated with mortality. And then I tried to see how well did they discriminate the LCA phenotypes. And so the plot on the left is showing factor values per patient sample. And I've colored and separated them by LCA phenotype. So as you can see, factors 2 and 3 seem to separate the phenotypes a little bit better than factor 1. And in fact, when you combine factor 2 and 3, we are recapitulating the LCA phenotypes pretty effectively. I then performed gene and metabolite set enrichment analysis on these factors to determine what pathways were involved in each. And in the interest of time, I will review the pathways, the factors specifically that I mentioned recapitulated the LCA phenotype, so factors 2 and 3, because those were also the factors that were associated with mortality. And so we see this sort of a coordinated cellular response in severe ARDS. On the left is the gene expression pathway analysis, and on the right is the metabolite enrichment analysis. So there's a decreased G-alpha signaling gene pathway. This pathway is crucial for generating cyclic AMP. So G-alpha proteins typically inhibit cyclic AMP production and dampen cellular response. So perhaps the fact that this is negatively enriched suggests that the cells are trying to increase cyclic AMP production to support enhanced cellular response, potentially in time of stress. And on the metabolic side, we see, again, increased fatty acid metabolites that suggest mitochondrial stress, again, those dicarboxylic fatty acids that are generated through omega oxidation. And then a lot of decreases in lipids that are associated with potentially membrane remodeling, maintaining barrier integrity. And also lipid signaling, so lipid mediators of inflammation, perhaps. But altogether, rather than some random degradation pattern, it seems to suggest that cells are potentially trying to rewire their metabolic response to support perhaps membrane integrity, membrane restructuring. Factor 3, again, gene set enrichment on the left, metabolite enrichment on the right. This factor revealed this sort of fascinating pattern that seems to suggest that there's activation of the integrated stress response, and that sort of fits with the metabolic changes that we're seeing as well. So on the left-hand side, we see increased activity of one of the key ISR pathways, and this is this EIF2 alpha K, and its translation-related processes, as you can see, and the other pathways represented. And this suggests that perhaps cells are activating stress responses and trying to maintain selective protein synthesis, again, during this critical illness. The right panel shows some of the metabolic consequences of that, or metabolic associations of that. So increase in polyamines, perhaps to shuttle sources to nucleotide synthesis. And again, sort of increase in lactated amino acids and drop in some of the same metabolites and acylcarnitines, in particular, long saturated acylcarnitines, that suggest that perhaps there's mitochondrial impairment or mitochondrial stress. And then finally, we honed in on the mitochondrial metabolites themselves. So there's, we used a set of metabolites that are known to be altered in plasma of patients with mitochondrial genetic disorders, such as MELAS, and found that nearly all of those metabolites were altered in our data set and elevated in the hyperinflammatory patients. And then we performed gene set enrichment analysis using specifically a mitochondrial gene set called mitoCARTA. And we found that there was increased expressions of key mitochondrial genes, and particularly oxidative phosphorylation, and including complex 1 and complex 3 subunits, and basically supporting or trying to increase the cell's ability to generate energy and maintain energy production during this time of stress. And so I'll focus a little bit on the integrated stress response before I finish. This is, for those of you who don't know, an evolutionarily conserved intracellular signaling network that helps the cell and the tissue and the organism to adapt to a variety of stressors and maintain protein homeostasis. And cell stresses can be quite variable, but include infections, mitochondrial stress itself, or dysfunction in any way can activate the ISR to sort of reprioritizes the cell's protein synthesis towards production of stress response proteins, and also helps the cell metabolically buffer itself against oxidative stress. And there's this idea that potentially too much activation of the ISR or too little activation can be maladaptive and lead to loss of cell fitness or organismal death. And so putting all this together, perhaps what we're seeing here is that in a subset of patients with hyperinflammatory ARDS, the pathways that seem to be associated with mortality seem to suggest that there is this enhanced maladaptive ISR response. There are limitations to what I showed you. All these interpretations are based on global data from circulation. So whether metabolites are altered because of a decrease in their production or an increase in their removal is unknown. We can't really answer that with the data that we have. And similarly, we don't know which cells or organs are generating these signals. We also don't know, I've chosen to split the heterogeneity question by the LCA phenotypes in ARDS, but we know that there are multiple different ways of defining heterogeneity in ARDS. An important contributor to heterogeneity, especially in metabolomics, are pathogens, and we don't have that data in this data set. There was actually a great paper that was published earlier this year by Dr. Winston's group in critical care that used targeted metabolomics, and they found that there were metabolic signatures that differed based on whether patients had COVID-19 or H1N1 or culture-positive bacterial pneumonia. So I think having that data would likely explain some of the heterogeneity that we're seeing. And then lastly, the patients in our analysis were selected on purpose to represent extremes of phenotypes. So potentially it's a sort of application of what I have suggested here to patients who fall sort of in the middle of that spectrum of hyper to hypoinflammatory phenotype is unclear. So in conclusion, molecular phenotypes of ARDS demonstrate distinct metabolic profiles. The hyperinflammatory phenotype seems to be characterized by altered mitochondrial function, impaired fatty acid oxidation, and activation of the integrated stress response, which may be a maladaptive response in this context. And these pathways seem to be significantly associated with mortality. And so they open new questions about whether targeting them could improve outcomes in severe ARDS. And I'd like to thank Lucille Naton, who is one of the bioinformaticians in our lab group and helped me with a lot of these analyses, and my mentors, Dr. Carolyn Calfee, Dr. Matthay, and Dr. Rogers, and the Calfee-Matthay lab, who supports many, many of our studies, including what I showed you today. And I'm happy to take any questions.
Great. That was a wonderful presentation by both Dr. Matthay and Dr. Alipanah-Lechner. We are going to take a few questions. If you have questions, please put them in the Q&A section. Before we get to that, I just want to put in one plug from our sponsor, Dr. Melissa Bates, and LSF Solutions is going to be starting a clinical trial with a device used to evaluate lung heterogeneity, which fits in well with the theme today. So you can contact Melissa. She left her email address in the chat, in the Q&A section. Okay, so the first question that I saw, I think maybe this is, well, either of you can address this one. This is from Ian Davis. Are there differences in alveolar fluid clearance rates between subjects with hypo and hyperinflammatory ARDS?
Hi, Ian. Thanks for the question. We don't know. We haven't had the capacity to measure alveolar fluid clearance in those patients. We would speculate it should be reduced in hyper, but we don't actually know.
And the next question is from Brent Winston. If you did not pre-select the hypoinflammatory phenotypes and use PCA analysis, did you see grouping of metabolites into clinically different groups?
Yeah, that's a great question. So that is what Dr. Rogers had done. She actually did not use LCA phenotypes. She just used metabolic data and found that they did fall into these three distinct groups with clinical differences, including mortality that was different. But in my second analysis that I showed you with transcriptomic and metabolomic data as well, that multiomic analysis also did not have LCA data. So it was just metabolites and gene transcripts, and sort of the model itself clustered them or recapitulated the phenotypes essentially, which is interesting because the phenotypes are defined using proteins. In some ways, it's nice to see that all these different modalities are capturing the same signals.
So this is a more fundamental question, maybe for some of the physiologists in the group, and maybe Michael can address this one. It's from Asfan Zahar. Can you explain the main difference of high stretch and low stretch ventilation?
Well, I would say, Afsan, that the biggest issue is that with the high stretch, the traditional way in which patients were ventilated with the higher tidal volume, higher airway pressures, the injured alveoli are just very delicate. And with this higher pressure, it results in biomechanical injury to the alveolar epithelium, endothelium, and a lot of data showing macrophages, for example, increase their pro-inflammatory factors. So I think it's a biomechanical effect, and the low stretch reduces that.
The next question is from Eric Morel. Narges, this was a great talk. Is there any animal data or mechanistic studies that might help us understand the organs and or cells most responsible for many of the metabolites you're measuring in the plasma? Do you think most of the metabolic differences between hypo are from the lung or elsewhere?
That is a great question, and an active area that our group is trying to answer next. We're in the process of doing some mouse studies to figure out what organ is the most responsible for this signature. I think from what I have reviewed, it seems that the liver and the kidney are probably two of the prominent sources. contributors to these differences. And I'm a little bit biased more towards the liver, I think, from some of the recent analyses I've been doing. I do think the lung is also a contributor, but I don't think it would be the primary contributor. But that's a great question. We will find out hopefully.
Well,
soon.
and just a comment, because I have some interest in this myself. I think there also when you get to the point of looking at animal studies or human lung tissue, I think that there's probably going to be heterogeneity both also spatially in the more injured regions versus the non-injured regions, and that might make it even more complicated.
Absolutely, absolutely.
Next question is from Atefa Mohammadi. Thank you for the great talk. I had two questions. I'm studying neonatal ARDS, and I'm wondering if much is known or studied about the different phenotypes and subphenotypes of neonatal ARDS. And then the second question is, I also study the role of arginine and citrulline in lung pathobiology and as a treatment for lung disease in neonates. So I'm curious about whether you have conducted any amino acid panels and whether you have noted any differences in the phenotypes there.
Well, I'll just take the first part briefly. Neonatal ARDS is a little bit beyond the scope of our focus, but I work with some neonatologists. There's a terrific investigator at Penn. Her name is Krithika Lingappan, Lingappan. She has dynamite work in this area. She and I just wrote a commentary, and I would definitely refer you to her, Krithika Lingappan. She's just terrific. I don't think they've done the formal kind of subphenotype analysis we did, but very relevant. Which premature babies get bronchopulmonary dysplasia and all of that. But let Narges tackle the second part.
Yeah, thank you for that question. We did include amino acids, and they are significant differences between the two classes across most amino acid types. Arginine and proline metabolism is elevated in patients with hyperinflammatory ARDS in this data set. But so it's all the other amino acid classes. So again, this is data from circulation, hard to interpret how it translates to what's happening within each tissue itself, whether sort of it's a signal that everything is getting mobilized towards energy production, or it's actually being taken up by specific tissues in different ways. It's hard to say.
Next question is from Mary Dahmer. Are there any potential therapeutic targets uncovered by your MOFA analysis which already have drugs approved for other uses?
Yeah, that's a good question. I think I would be cautious in wanting to act on this data yet. Again, I think not knowing where the signal is coming from, or is this truly sort of are the signals we're seeing the cells attempt at helping survive this condition, and by modifying it, we would inadvertently cause more harm, is not clear from this data. But I would say the integrated stress response, I think, has known modulators both for overexpression and underexpression. Unfortunately, it's such a widespread pathway that most cells in the body utilize. So I think there's a lot of side effects with the use of those drugs. But none that have been studied in critical illness and in this context. So that's an area that hopefully will be investigated if these pathways that we're discussing turn out to be causes that are contributing to death in these patients.
Okay, so we're going a little over. We have time for maybe one or two more quick questions. Let's see. Let me go to... there's a question here from Jerome Dempsey. Do you think lung mitochondrial function is a major source of the mitochondrial dysfunction?
Yeah, that's a great question as well. I think there's some data by a group from Yale that did a comparison of different tissues and how potentially infection impacted to metabolic changes in these different tissues. And it seemed that the liver and the kidney were the primary sources of heterogeneity, but there is definitely compelling evidence also from the lung. Which one is dominating what we're seeing in circulation? I think it's probably, my guess would be it's the liver and the kidney. But I think what is contributing to respiratory failure and respiratory failure severity would probably be more observed in a lung-specific study.
Yeah. Okay, last question. Untargeted metabolomics from Brent Winston. Untargeted metabolomics can only take you so far. Are you planning on repeating analysis with targeted metabolomics?
Yes, that is a great question, and a huge limitation of untargeted metabolomics is that you can compare within a data set, but not across studies. And also, we don't really know the absolute quantities of any of the metabolites that we're measuring. Our hope is that if we can sort of replicate these findings in our mouse model to then go for a more targeted approach to a select few of these metabolites that we think are most representative of this signature that we're observing. So yes, that is our plan.
Okay, I'm sorry I didn't get to some of the other questions, both in the chat and in the Q&A, but we are going to have to wrap up. I want to thank our sponsor, LSF Solutions, and Dr. Melissa Bates once again for supporting this. The respiration section for organizing it, APS for supporting this, Dr. Raj for an excellent introduction of our speakers, and finally our speakers themselves. Thank you so much. That was really wonderful. Thank you all.
Thanks so much.